Okay, today we are going to discuss about the new topic from your syllabus, that is synthetic antibacterial agents. In your syllabus, it was mentioned like synthetic anti-infective agent. So anti-infective, antibacterial one and syn. So it includes the sulfonamides, then folate reductase inhibitors, quinolones, and miscellaneous compounds, that is unclassified compounds like furazolidine, nitrofurantoin. Actually, this nitrofurantoin belongs to the class nitrofuran because it contains the substituted furan ring, that is nitrofuran. 5-membered oxygen containing ring is furan, and NO2 group is there, so it becomes nitrofuran. And nitrofurantoin is the one of the example from that class, that is nitrofurantoin. And methenamine, so another unclassified or miscellaneous agent. So it's a very interesting and simple chapter, that is synthetic anti-infective agents or synthetic antibacterial agents. So one and the same: sulfonamides, quinolones, and nitrofuran plus nitrofurantoin. See, we had already discussed about the various antibiotics, right? Like beta-lactam antibiotics, penicillin, cephalosporin, streptomycin, aminoglycoside antibiotic, tetracycline antibiotic. See, some of them are the bactericidal or some of them are the bacteriostatic in nature. Means some antibiotics have the ability to inhibit the growth of bacteria, or certain antibiotics have the ability to directly kill the bacteria. So some of them are bacteriostatic or some of them are the bactericidal. That is antibiotics that is naturally obtained from either plant sources, animals, microorganisms, so and so, right? But what about the synthetic antibacterial thing? These agents work against the bacteria. They act against the bacteria, either inhibit the growth of bacteria or directly kill the bacteria. So depending upon their potency, again they are classified into two, that is bactericidal and the bacteriostatic. Bactericidal means those have the ability to kill the bacteria, and bacteriostatic means those have the ability to inhibit the growth of bacteria. Now the word synthetic means these are the agents which are synthesized in the laboratory. This agent works against the bacteria, either inhibit the growth of bacteria or kill the bacteria, but they are not obtained from the natural sources like plants, animals, or microorganisms, or etc. They are synthesized in the laboratory, right? Synthetic, that's why they are called as synthetic antibacterial agents. Means other than the antibiotic, these are the substances, these are the chemical agents which have the ability to kill or inhibit the growth of bacteria, but they are synthesized in the laboratory. Now, what are the examples of synthetic antibacterial agents? See, these are the main or typical examples of synthetic antibacterial agents like sulfonamides, quinolones, and nitrofurans. Sulfonamides, quinolones, and nitrofurans. So these are the main classes belong to this synthetic antibacterial or synthetic anti-infective agent, sulfonamides. Again, number of drugs comes under the heading of sulfonamides. Number of drugs are there in the quinolones. So these are the two important classes from this synthetic antibacterial agent. Sulfonamides, say for example, sulfacetamide, sulfaguanidine, sulfamethoxazole, etc., etc. These are the examples, or these are the drugs belongs to sulfonamide. Quinolones, say for example, ciprofloxacin, norfloxacin, gatifloxacin, moxifloxacin. These are the drugs which belongs to the quinolone class. And nitrofurantoin is an example of the drug which belongs to this nitrofuran class, right? See, first we'll discuss about the sulfonamides. Now, what exactly the sulfonamides? Sulfonamides are the bacteriostatic in nature and are effective against many gram-positive and the gram-negative bacteria. Particularly, they are the bacteriostatic in nature. They are not the bactericidal. Sulfonamides are the bacteriostatic in nature and are effective against gram-positive and the gram-negative bacteria, and they are used in the treatment of UTI, that is urinary tract infection, bacillary dysentery, then burns, malaria, conjunctivitis, nocardiasis, streptococcal infection, gum infection, and the meningitis. Then history of sulfonamides. Actually, the sulfonamide story began in the year 1935 when it was discovered that a red dye, that is prontosil. So this is the prontosil, and this prontosil was used as a dye in the dyeing industry at that time. In the year 1935, it was discovered that a red dye, the name of that red dye is the prontosil, and this is the structure of prontosil. So in the year 1935, the red dye, the name of that dye is the prontosil, and that prontosil had the antibacterial properties in vivo, that is when given to laboratory animals. See, in vivo and in vitro. In vivo means inside the body, right? In vivo, inside the body, and vitro means outside the body. In vivo means inside the body, and in vitro means outside the body. So in the year 1935, it was found that the red dye, that is prontosil, this red dye prontosil, had the antibacterial properties in vivo, that is when it is given to the laboratory animals. But strangely enough, the no antibacterial effect was observed in vitro, that is, it does not show any antibacterial property outside the body. In vitro means on your experimental culture, on your microbial culture, right? So there was no antibacterial effect was observed in vitro. We can say that, in other words, prontosil could not kill the bacteria grown in your test tubes, right? And this remains a mystery until it was discovered that the prontosil was not in fact the antibacterial agent. So instead of this prontosil, it was found that this dye was get metabolized by the bacteria present in the small intestine of the test animals, that is the mice, and after metabolism, this prontosil is broken down to give the metabolite, that is the sulfanilamide. After metabolism of prontosil, we'll get sulfanilamide or the sulfonamide. Means, in other words, we can say that prontosil is the prodrug of sulfonamide. Sulfonamide, see, all sulfonamides are the derivatives of sulfanilamide. See, basically this structure NH2, benzene ring, SO2 and NH2. Basically, it is called as sulfanilamide. This is the structure of sulfanilamide. When you replace either one or two hydrogen, at that time you will get the derivatives of sulfanilamide, and that derivatives are classified under the chemical class sulfonamide. Yes, so prontosil is the prodrug of sulfonamide. Prodrug, basically prodrug is the inert, right? Prodrug is the inert, that is inactive form, and it is get activated after the metabolism. See, this prontosil shows antibacterial properties in vivo, inside the animal, because basically prontosil is the prodrug, and it get metabolized by the particular enzyme, and that enzyme is nothing but the azoreductase, right, which break this bond, N double bond N. Its history, it started in 1935 when it was discovered that the prontosil, the red dye, shows antibacterial properties inside the body, that is in vivo, but there is no antibacterial effect was observed in vitro. Means you can say that prontosil could not kill the bacteria grown in the test tube or the predictive plate. So this is the mystery until it was discovered that the prontosil was not in fact the antibacterial agent. So instead, the prontosil is the prodrug, and it gets metabolized to its active form, that is sulfonamide, and that sulfonamide has the antibacterial properties. So next, about the physicochemical properties of sulfonamide. So different physicochemical properties we'll discuss, that is solubility, reactivity, antibacterial potential, pH and pKa. So what about the solubility of sulfonamides? Sulfonamides are all white crystalline powders and mostly they are poorly soluble in water. So remember this. They are white crystalline powder and poorly soluble in water. Their solubility is very poor in the water, and their sodium salts are usually used because of its low aqueous solubility. So to improve the solubility of the any compound, we need to prepare the salts of that compound, right? So all sulfonamides are generally white crystalline powders and they are poorly soluble in water. So to avoid this problem, generally the sodium salts are usually used because of their low aqueous solubility. And the solubility parameter is generally influenced upon by the nature of the substituents which are present on the SO2NH2 group. See, the solubility of any moiety which belongs to sulfonamide, it totally depends upon the substitution which is present on this SO2NH2, means particularly the substitution which is present on the nitrogen atom, sulfonamide moiety, right? Then what about the reactivity? So substituents which are present on the SO2NH2 group again modify the chemical features of the molecule. Means total the reactivity of the compound, it depends on this group SO2NH2. What about the antibacterial potential? Sulfonamides are bacteriostatic in nature, but in the very rare cases where the bacteria are exposed to the thymine-less medium, then in that case sulfonamides act as the bactericidal. Instead, most of the sulfonamides are the bacteriostatic in nature. Then antibacterial potential of sulfonamides are gradually decreases by the presence of pus, tissue fluids, and such drugs which contains PABA as the basic skeleton, that is para-aminobenzoic acid. And what is the example of the drug which contains PABA as the basic skeleton? For example, local anesthetic. Antibacterial potential of sulfonamides are gradually decreases by the presence of pus, right? Then tissue fluids and such drugs which contain PABA as a basic skeleton, for example, local anesthetic. So that pus contains large amount of the PABA, para-aminobenzoic acid, as well as the local anesthetic or any drug which contains PABA. In presence of that drugs or in presence of the pus, the antibacterial potency of the sulfonamides will decreases. Then what about the pH and pK? The importance of the pK values in designing the effective sulfonamide antibacterial. The active form of sulfonamide is the ionized form, and maximum activity is observed between the pK values in between 6.6 to 7.4. So these are the some physicochemical properties of sulfonamide: solubility, reactivity, antibacterial potential, pH and pK. And next point is classification of sulfonamides. See, this is the general structure of sulfonamide. See, how to draw it is very easy. See, draw the benzene ring NH2, then SO2NH2, sulfonamide group. See, benzene ring NH2, SO2NH2. And what is the name of that structure? Sulfanilamide. SULPHANILAMIDE. Sulfanilamide. But when you replace one or both the hydrogens by any substitution, then the name of that compound becomes sulfonamide. That comes under the class sulfonamide. Sulfonamide is the class, and all sulfonamides are the derivatives of sulfanilamide. Sulfanilamide, or you can say sulfanilamide. See, basically it's a sulfonamide. Sulfanilia, benzene ring NH2, anilia, sulfanilamide. But when you replace the hydrogens, either one or two, from this nitrogen, so that becomes the general structure of sulfonamides. Basically, all the sulfonamides are the derivatives of sulfanilamide. Now, they are classified according to the duration of action and according to their use. See, sulfonamides are classified according to the duration of action and use. So, based on the duration of action, they are classified into short acting sulfonamide, intermediate acting, and long acting. And according to the use, they are classified into sulfonamides for ophthalmic use and sulfonamides for burn therapy. So according to the duration of action and according to the use, sulfonamides are classified into, according to the duration, they are classified into three classes: short acting, intermediate acting, and long acting. According to the use, sulfonamides are classified into sulfonamides for ophthalmic use and sulfonamides for burn therapy. See, if you want to draw these structures, each and every structure from this chapter, sulfonamide, you just remember this basic skeleton. NH2 benzene ring SO2 NHR. Just remember this one. NH2 benzene ring SO2 NHR. We will discuss one by one in detail: short acting, intermediate acting, long acting, sulfonamides, etc. So first class from this, short acting sulfonamides. See, why they are short acting? Because these sulfonamides are rapidly absorbed and rapidly excreted, right? Rapidly absorbed in the body and rapidly excreted. That's why they are short acting sulfonamide. Their half-life is generally from 4 to 7 hours and they are administered every 4 to 2 hours. They are short acting. Why they are short acting? Because they are rapidly absorbed in the body and get rapidly excreted from the body. Now what are the examples of short acting sulfonamides? Yes. First example is the sulfoxazole. See, it is called as sulfoxazole. See what that indicates? Sulfoxazole. Yes. The name of that compound, in the most of the books it was mentioned like sulfoxazole. Sulfoxazole, but I deliberately write like this: sulfoxazole. So it is very easy for me to remember the heterocyclic ring which is present in the sulfoxazole, right? See, sulfoxazole, or in simple way you can call it as sulfoxazole, but it is very easy to remember the heterocyclic ring. Which heterocyclic ring is present in the sulfoxazole? That is sulfoxazole ring. So see, the structure is large, but it is very easy to remember. Draw the general structure: NH2, benzene ring, Sa2, and NH. Correct? See, I will put all the structure over this: sulfoxazole, then second one is the right sulfamethiazole, then sulfapyridine, sulfamerazine, and sulfadiamidine or sulfametazine. So these are the some examples of sulfonamides, short acting sulfonamides. You need to remember at least one structure from this class: short acting sulfonamide. Sulfoxazole, sulfamethiazole, see, sulfoxazole, sulfamethiazole means heterocyclic ring, thiazole. Basically it's a thiadiazole. Then third compound is the sulfapyridine. So which heterocyclic ring is present over here? Pyridine ring. Then sulfamerazine. So identify the heterocyclic ring which is present in sulfamerazine. Yes. Here if you observe, it's a 6-member unsaturated ring and 2 nitrogens are there. But and what about the positions of that nitrogen 1 and 3? So that becomes pyrimidine. If the 2 nitrogens are there in the 6-membered ring and the positions are 1 and 2, that becomes pyridazine, right? When 2 nitrogens are present but the positions are 1 and 2, that becomes pyridazine. In position 1 and 3, that becomes pyrimidine or pyrimidine. This is pyrimidine. And what about this sulfadiamidine or sulfametazine? Which heterocyclic ring is present over here? Again it is pyrimidine. So how to remember all these structures? See, I will mark the heterocyclic ring which is present in every compound. Sulfoxazole. So this is my isoxazole. It's not the plain, it's a substituted isoxazole ring. All are the substituted except sulfapyridine. Then second one is sulfamethiazole. So if you observe, see, sulfamethiazole or thiazole. Is it thiazole? No. The name of that compound is thiadiazole because 2 nitrogens are present. Thiadiazole. So this is thiadiazole, substituted thiadiazole. In case of sulfapyridine, pyridine ring is present. So see, it is very easy to draw the structure: sulfapyridine, because pyridine ring, it's a plain pyridine ring. Next, sulfamerazine, pyrimidine ring is present over here, and again sulfametazine or sulfadiamidine, pyrimidine. See, the marked portion from all these compounds are the heterocyclic ring, or you can say that is R, right? See, Remember the general structure of our sulfonamide. NH2, benzene ring, SO2, NH and R. So this marked portion is nothing but the R, right? So you just remember the R moiety which is present in all these compounds. The rest of the skeleton is same. NH2, benzene ring, SO2, NH. NH2, benzene ring, SO2, NH. NH2, benzene, SO2, NH. NH2 benzene, SO2 NH and NH2 benzene, SO2 NH. See, rest of the moiety, this is our parent, right? Rest of the moieties are common for all these structures. But what is the difference between all these drugs? Yes, the presence of the heterocyclic ring. So R is, see, in case of sulfa-isoxazole, R is isoxazole ring, sulfa-methiazole, R is sulfa-diazole. For this sulfa-pyridine, R is pyridine ring. For sulfa-merazine, it's a pyrimidine ring. And again, the pyrimidine ring is present in sulfa-diamidine. Then what about the IUPAC name of all these compounds? See, this is our parent, right? Benzene sulfonamide. This is our parent. So, benzene sulfonamide. And this parent compound, this parent skeleton is present in all these derivatives. So, the parent is benzene sulfonamide. Now, numbering starts from this. See, benzene sulfonamide. This is our parent. Now, numbering start from this for this benzene ring. 1, 2, 3, 4. And one substitution is present at the fourth position on the benzene sulfonamide. That is amino group. So, name of that compound becomes 4-amino benzene sulfonamide, right? 4-amino benzene sulfonamide. When this heterocyclic ring is absent, that becomes NH2. And the name of that compound is 4-amino benzene sulfonamide. So, this 4-amino benzene sulfonamide is parent in all the structures. Sulfa-isoxazole, sulfa-methiazole, sulfa. Right? This is 4-amino benzene sulfonamide. Again, this is 4-amino benzene sulfonamide. Again, this is 4-amino benzene sulfonamide. Now, how to write the IUPAC name or the chemical name for this structure? These are the common names, sulfa-isoxazole. So, how to write the IUPAC name or the common name? See, my parent is benzene sulfonamide. Again, one substitution is there at the fourth position on that benzene sulfonamide, that is NH2. So, I write like this, the benzene sulfonamide, one substitution is present on benzene sulfonamide with respect to position number 4. Which substitution is there? That is amino. So, 4-amino benzene sulfonamide. Again, one more substitution is there on the nitrogen. So, I write like this, N in bracket. N in bracket. Now, for this is isoxazole ring, right? So, numbering start from which? This, oxygen. So, 1, then 2, 3, 4 and 5. 1, 2, 3, 4 and 5. That is 3,4-dimethyl. 2 methyl substitutions are present on isoxazole ring. So, that becomes 3,4-dimethyl isoxazole 5-il-C. This isoxazole ring is attached to parent with respect to position number 5. So, name of that compound becomes 4-amino N in bracket 3,4-dimethyl isoxazole 5-il benzene sulfonamide.